that we will discuss it in detail a little bit. And also, we should be able to identify or analyze the basic characteristics such as size, granularity, and the expression of a specific gene or protein in the cell. And also, we can use flow cytometry to check the expression level of biomarkers, and we can quantify the existence or the expression level of biomarkers compared to a control means, for example, if we have two groups two groups of cells, one group of cells you treated with a compound expecting that that compound is having some anti-inflammatory effect. And then when we plan this experiment and do a flow cytometry analysis for the biomarker of your interest, you can compare the expression level with your treatment group versus control, and that will give you a quantitative analysis. And also in advanced flow cytometry techniques, we can use several antibodies which can be fluorescently tagged. That antibodies The antibodies which we used can be tagged with a fluorochrome. Fluorochrome is a compound which excites and emits light of a particular wavelength. So based on that excitation emission frequency, we can tag these antibodies. The antibodies will go and bind to the antigen or the protein of our interest. That will give a quantitative analysis. And there are several multicolor system available. As far as I know, we can detect, there are flow cytometry techniques available to detect more than 30 parameters simultaneously within a single cell. So these are highly emerging as well as advanced technology which is being operated in several sort of flow cytometry applications. And also, flow cytometry is useful for the identification, purification, as well as sorting. Cell sorting is very, very, very important. For example, if you check our blood cells, WBCs, those are heterogeneous population. You will see lymphocytes, neutrophils, macrophages, monocytes like that. So that's a heterogeneous population. And if you want to specifically sort or isolate a specific set of subset or set or subset of cells, flow cytometry can be useful. And looking into the components of flow cytometry, because why as a medical student or a medical professional we need to know about this, because if we know the fundamentals, if we know the basic principles of any instrument, that will help you to troubleshoot if you come across with any issues. So flow cytometry, this is very basic thing. We can see in flow cytometry, it consists of three different components. First one is fluidics, optics, and electronics. Three components: fluidics, optics, and electronics. Optics, as we know, anything related to light. So if you see the picture one, there will be a tube. That tube is called flow tube. The cells will be passing through that flow tube as single cell suspension. So one cell at a time. So we have an optics, that means an optical system which can illuminate the cell using a laser light. So a laser beam will be illuminating the cells, and that will trigger, if the cells are tagged with a fluorochrome, that will trigger fluorescence. If they are just untagged cells, raw cells, it can scatter the light. So there will be a detector to see how much it is scattered or how much fluorescent signal is being emitted. That will be captured by the detector and converted into to digital signals. So that's how the optics work, and there will be filters available to select appropriate wavelength. For example, if you use a fluorochrome which is emitting a green, so that means it should be excited by a blue light. So blue light, it excites and it will emit green light. So like that, there will be excitation emission profile for each of the fluorochromes. Accordingly, we need to choose the light source. So So those appropriate wavelength selection will be done through filters. And there is an electronic system mostly composed of a PMT. PMT is photo multiplier tube. So photon is the light, so it can amplify the signals and convert it into a digital computer display, which will be useful for analysis. And another important component is fluidics, which consists of a flow chamber. So in a flow chamber, the cells will be aspirated through a small capillary tube, and these cells will be passing through a flow tube, which is a very small sized micrometer diameter sized tube, so that cells can only an individual cell can pass through it. So that's how we can get the real-time data available on a single cell basis. And when we look into flow cytometry, two important parameters you need to consider. One is forward light scattering and another one is side scattering. So if we are using a cell which is untagged, which has no antibodies conjugated or which has no fluorochrome conjugated with any of the molecules. So it's an untagged cells. When it flows through the flow tube, the laser will be hitting to that cell, and that will give two types of scattering. One set of scattering called forward scattering, where the light will be scattered towards the forward direction. If you see this picture, you can see the light path is coming and it is hitting the cell. And if you look into FSC, for forward scattering, the scattered light which is moving towards the forward direction will be captured. So this will give an information, an idea regarding the size of the cell. So forward scattering means that is related to cell size. Because larger cells can scatter more light, smaller cells can scatter lesser light. So accordingly, it will calculate the size of the cell. And if you look into the principle, the scattering is primarily due to the diffraction of light around the cell surface. So the idea here is forward scattering will give you the size of the cell. And how about the side scatter? Side scatter, here the light will be scattered across the cell, mostly around 90-degree angle relative to the laser beam. So here the cells will give the information which we obtain would be internal complexity or granularity of the cells. If the cells contain too much granules inside, for example, if a cell with too much mitochondria or too higher plasmic reticulum or something like that, that granularity information will be given by side scattering, which includes the presence of organelles or density of organelles, granules, and other structures. Here, side scattering, when the intensity is higher, it will give more internal structures. So the takeaway here is forward scatter means it will give the size of the cell. Side scatter means it will give the granularity of the cell or the internal complexity of the cells. So that's what we need to understand when we look into flow cytometry, especially when we do all experiments, we need to do an untagged experiment to see the forward scattering and side scattering. That will give an idea regarding the size and granularity of the cells. Okay, so these forward scattering and side scattering can be used for gating the cells. Gating means if we want to isolate a particular population of cells, we can use gating system. I will be explaining gating in the forthcoming slides. Okay, so when we do forward and side scattering, the system will create a scatter plot. You can see the forward scattering on the x-axis and side scattering on the y-axis. So four different population of cells are here. The first one is cell debris. You can see small, incomplete cells or cell aggregates. Everything will fall over here. And you can see lymphocytes in this region where it is more towards forward scattering. Above that, you can see monocytes and granulocytes all over. You will see over this large bubble. So that means based on this forward scattering and side scattering, that itself will help us to identify and sort different cell population. Okay, considering this every time, so lymphocytes are of very small, so it will appear initially in the forward scatter, whereas granulocytes, the other WBCs like monocytes and others are bigger in size compared to lymphocytes, that will be appearing more towards granular side. So this is a simple example. If you do flow cytometry using any instrument or anywhere, this pattern should be reproducible. So this is more or less a standard pattern for doing flow cytometry sorting for uh, WBCs. And now you may be coming across with another terminology called FACS, fluorescence activated cell sorting. Again, here also, every time consider when we do any cell experiment. For example, if you isolate stem cells, or if you isolate blood cells, or if you isolate any type of cells from any tissue, or if you give a treatment to a certain set of cells, these cells behaves heterogeneous. We will be thinking those are of same phenotypes, but technically they are heterogeneous. That means there would be a mixed population of different cells. So in order to purify that, we need to know the signature of each population. Okay, for example, lymphocytes can be T lymphocytes can be sorted based on TH 8 positive cells or TH 4 positive cells. Like that, we need to have some signature molecules which are called biomarkers available for these cell population. So based on that, we can design antibodies with some fluorochrome attached to that antibody. So when we use that antibody to bind these cells, it will be it will be binding with the cells. At the same time, upon that particular laser, it will be emitting some fluorescence. So based on that fluorescence, we can detect and sort the cells accordingly. That process is called fluorescence activated cell sorting, or FACS. Here, this FACS based approach is mainly useful for detecting the gene expression. We can quantify the expression of genes, and from a heterogeneous population, we can also identify what percentage of the cells give a positive expression for the molecule of interest. Here also, you can see, say, simple diagram. You can see there is an antibody conjugated with a green fluorochrome, and there are some cells which has the black dots represent the unconjugated molecules. So if you see this antibody, the cells will be receiving this antibody. The antibody will be binding with the cells, and that will give a green fluorescence. And when excited with the laser, based on that green fluorescence, the detector will choose only green fluorescence and take those cells to a container. So here the untagged cells, that means the black one, should be taken to a different compartment. So that's how it works. So the fluidics system will detect the presence of an fluorescence, and then that fluorescence will be used as a separate sorting criteria, where the cells will be taken into a sorted compartment. All those cells which are not positive for that fluorescence will be taken to a different compartment. So likewise, we can design multiple fluorochromes. So accordingly, depending upon the complexity of the machine, it will be helping us to sort as many cells as possible. Yeah, when we look into the working principle, how does it work? As we mentioned, three major components: optics, electronics, as well as the detector. So when you look into the overall arrangement of a flow cytometer. You can see a chamber where we can give cells. Okay, you can see a beaker with different types of cells tagged with different antibodies. We have a type of cells with antibody A and B. We have another set of cells with only one antibody, another one with a different antibody, and we have unstated cells. So what the machine does when we start the machine, when we start the program, it aspirates the cells, and there is a ultrasonic nozzle vibrator that will help to separate the cells into single cell suspension, and it will be passing through the flow tube when the laser is hitting to that molecules. According to the expression of those proteins, these cells can be sorted. So we will get four different population of cells which is negative for A and positive for B, double positive, double negative, as well as positive for A and negative for B. So in a simple experiment, we can sort four different types of cells. For example, if you are looking into two different antibodies, tag those two different antibodies with two different fluorochromes, and you will get four population of the cells. Four population of cells: antibody A, antibody B, double positive, as well as double negative. Okay, so another important avenue to look into is gating. So gating is simply it's the process of selecting specific subset of cells. For example, if you see this figure, this is the same figure as I mentioned before. So you can see the debris here, you can see the lymphocytes here, you can see the monocytes here. So just based on this dot plot, we can simply choose the region of cells of our interest. For example, if you are looking into only lymphocytes, just use the selection tool to select those area. So that will only pop up, that will only give those cells which are under our gate. So that is called gating. Gating is simply creating the boundary. When we create a small boundary like this inside this population, specific population of cells, that will select only those particular population which is within the gate. Gate is simply a selection tool that will help us to further identify the expression level and further sort those cells. So if you see the flow diagram here, so we have a population of WBC. We select only lymphocytes. You can see the gate here. So now we got only those cells which are lymphocytes, and then we need to do a subselection of other phenotypes. Like we need to separate B lymphocytes and T lymphocytes and NK cells. So which can be again further sorted. That will give three different quadrants. In quadrant 3 gives T lymphocytes. Okay, we can select again quadrant 3 that will give the subset of T lymphocytes that are CD4 and CD8 cells. Okay, if you want to select the subpopulation of CD8 cells, just select the CD8 cells and do one more. So likewise, we can simply sort gate, that will give our subpopulation of interest. So this is very important in flow cytometry. That means we need to understand which population needs to be gated. We need to have a thorough idea of what all molecular markers are involved, and we need to know how to do specific set of gating in order to avoid contamination. So when we do gating, if we are not doing a proper gating, it may cause contamination of other cells as well. Okay, so there are several softwares available to do the gating as well as analysis. FlowJo is one of the most widely used. I think we do have FlowJo software here. And also we have a CellQuest. These are third-party softwares which is specifically designed for flow cytometry based experiments. And what are the applications? There are so many applications for flow cytometry. We can do cell phenotyping. We already mentioned those. We can do cell cycle analysis. We should be able to see what phase of cells are there. For example, cell cycle include 4 different phases. In a population of cells, how many cells are in the S phase? So we can do experiments for that. We can do cell function. For example, if you want to see whether a channel is activated or not. For example, there are molecules available to detect the activity of calcium channels. So if calcium channel is active, there will be a fluorescence within the cells or outside the cells. So depending on those fluorescence intensity, we can analyze the cellular function, and it can be useful for microbial sorting. And also there are several AI based databases which is being integrated with the flow cytometry program. So wide application we can use. And also we should be able to detect small particles like nanoparticles, vesicles, could be sorted using flow cytometry based techniques. So that means the applications are immense. So it's all depends on how we understand the process and how we design our experiments and what data output we are required. Accordingly, we can design flow cytometry experiments. And you probably have already attended the lecture from Dr. Rai regarding omics. So still we know, I'll give you a very brief overview. Omics is, it include genomics, proteomics, metabolomics. So anything related to omics can be done through a flow cytometry workflow. So for example, simple, consider this situation. You treated an animal with a drug. You created a disease model. You treated the animal using a drug. And for example, you induced a cancer in mice, pancreatic cancer in mice. So you treated a compound, or you treated with a novel drug, and you want to see whether the effect is there or not. What we will do after the treatment phase, we will sacrifice the animal, we will harvest the tissue, we can isolate the cells, and we can use flow cytometry based sorting for using the specific set of biomarkers. And once the sorted cells are available, what you can do, either you can culture those cells and expand a cell line which is positive or negative based on the expression of the marker. So those sorted cells can be used for analysis. For example, if you want to see a specific type of cancer cell, excuse me, yeah, if you want to work on a specific type of cancer cell, or a premature cancer stem cell, so you just use the biomarkers, isolate the cells, use that particular biomarkers, sort only those premature cancer stem cells, use it for omics analysis. So that will give a clear picture of what is happening in those particular cell types. So if we use the heterogeneous cell types, we may be getting unwanted results, or our results may not be that much reliable, because what we are observing would be from a different population of cell as well. So during such situations, just when we do for omics analysis, we sort the cells based on the biomolecule of our interest and go for analysis. Yeah, here again, there is a workflow. If you look into blood-derived cells, we have already seen how gating and cell distribution in blood cells. And also, if you look into solid tissue, just isolate, make the single cell suspension, fix the cells. So this is the flow protocol. So if we have cells, isolate the cells, fix the cells, and go for staining with antibodies. Okay. And if you have an adherent cell culture, some cells will grow in the culture plates. So again, we need to take those cells out, make it a single suspension, fix it, and go for staining with antibody. Once it is stained, do flow cytometry, do gating, and go for data analysis. So again, the question here is, if you are sorting the cells just for data analysis, this procedure will work, because fixing and permeabilization will kill the cell. Okay, suppose if you want to sort a population of cells, and if you want to grow those cells, you need to avoid this step, because fixing means, or permeabilization means, it will kill the cells. So we need to avoid these cells if you want to further propagate your cells of interest. And again, this is more or less same as we mentioned before. Okay, this is the protocol, this is the workflow. You need to be very careful, because this is the standard workflow for any flow cytometry based techniques. So first of all, again, sample collection, either from tissue or isolation, or from the cultured cells or blood. Make it into a single cell suspension. Very easy, just suspend the cells in any physiological buffer. Then you need to do a blocking. Blocking, usually we use horse serum or bovine serum albumin. BSA or horse serum we use to block. Now, what is blocking? Blocking will prevent the unwanted binding of antibodies. It will mask all those antibodies which is non-specific. Okay, there could be some non-specific antibodies binding to each other. So the blocking will block all those, or mask all those unwanted antibodies. And then you need to take the cell surface markers if you want to do for live sorting. As I mentioned, if you want to propagate the cells or culture the cells further, go for live sorting. Then we will be continuously washing the unreacted antibodies, unreacted chemicals, all those things needs to be washed using any physiological solution. We commonly use PBS. And if you don't want to go with live cell sorting, we need to fix the cells with the formaldehyde or paraformaldehyde. And we need to permeabilize the cells. Permeabilization means, for example, if an antibody is cytoplasmic or nuclear. Okay, sorry, if the protein of interest is cytoplasmic or nuclear, that means the antibody with the fluorochrome, it has to cross the cell membrane, go inside the cell, and bind with the protein of our interest. Similarly, the antibody with fluorochrome conjugation should cross the cell membrane, go all the way to the nucleus, and bind to the protein of interest. So that process is called permeabilization. For permeabilization, we use some detergents, basically Tween or Triton. Triton is the most commonly used detergent for permeabilization, because Triton is non-ionic detergent which cannot do much harm for the cells. And then that will stain for the intracellular biomarkers. Again, we need to wash. Then we need to add the fluorochrome conjugated antibodies. Again, wash and analyze. So we need to have a cell suspension. We need to do blocking. We need to do extensive washing. We need to do fixing and permeabilization based on the analysis. If it is live cell sorting, we need to skip those steps. Otherwise, it will kill the cells. If your protein of interest is intracellular, that means inside the cell, that means inside the cytoplasm or nucleus or inside any organelles, we need to do permeabilization. That means we need to use detergent like Tween or Triton. Those detergent will make small pores in the membrane that will take antibody inside and bind with the protein of interest. So this fixation and permeabilization is one of the key steps. So again, the takeaway here is, if you are going with a live cell sorting, that will be basically based on the antigens or the proteins present in cell surface. So we can avoid permeabilization or fixing step. And if your protein of interest is inside the cell, you need to fix and permeabilize. Okay, so that's very, very important when considering flow cytometry based experiments. Again, this is another protocol. it's more or less the same. So if you are looking into extracellular proteins, okay, just dissociate the cells, stain with the antibody, no need to fix. Okay. If you are looking into cytoplasmic proteins, get the cells, fix it. We need to fix it. Do permeabilization. You can see small pores in the membrane, go for stain. And then if you are looking into nuclear protein, okay, cell dissociation, I mean cell suspension, fixing, permeabilization, and staining. The stain will go and bind with the antibody will go and bind to the nuclear proteins. So that's how the flow cytometry protocol works. And single cell RNA sequencing, I don't know how familiar you guys are with this technique. So this is one of the highly advanced technique which studies the unique genetic makeup and molecular characteristics of individual cells. Okay, so according to Human Genome Project, each cell will be having around 30,000 cells. So in this technique, what they do, we will be analyzing or quantifying the expression of around 30,000 genes in each cell. So if you take one cell, you will get 30,000 reads. You will get 30,000 hits. Close to 20 to 30,000 hits you will get. So likewise, if you go with maybe 10,000 cells, the recommended cell number is 5,000. So if you have 3 groups, maybe you will be going with 15,000 cells. From each cell, there will be 20 to 30,000 hits. So that will give a very bulkier data. So this protocol, this technique is integrated with flow cytometry. So flow cytometry, as we know, cell sorting, it can do individualized cell sorting. Based on that, the single cell RNA sequencing protocol work. So this is the workflow in order for you to understand, isolate or identify the cells, get the cells into single cell suspension. You can sort the cells based on their biomarkers of interest. And there is a program or there is a technique to barcode all the cells. Every cells will be barcoded. Then those cells will be sequencing based on the expression of cells in each genes in each cell. There will be clustering. All these things are automated now, so we no need to worry about anything. Just give the healthy ideal population of cells. The system will give all those data in a quantifiable as well as accessible form. Okay, the major applications include to identify gene expression at single cell resolution, to identify the cellular heterogeneity, and then for the cell type identification, we can pick the cells of interest and we can redefine or we rediscover new subpopulation. This is always helpful in disease diagnosis and treatment and also in developmental biology. So tremendous application has been associated with single cell RNA sequencing. So flow cytometry is an integral part where the single cell input is being provided by flow cytometry based techniques. And uh, that's all about the fundamentals of flow cytometry. I can give you some exercises if you want to try. Okay, anybody can volunteer. So for example, here we have 4 different problems to be identified. Okay, we have our first task would be isolation and sorting of Th17 subset from blood. Okay, second one, quantification of myofibroblast from a wound. Then CD90 plus heterogeneity of stem cells isolated from adipose tissue. And quantification of a GFP expressing cells after transfection. You can choose any one of this. And can someone volunteer and give an overview how we develop a protocol. You no need to specifically know any specific biomarkers. You can probably make it as X or Y. Christian, you want to try?
No,
Oh, yeah. Chisom, come on.
I can, yeah, let me see. So for um maybe if you wanted to quantify myofibroblasts from a wound,
Okay.
um since these are muscle cells, I'm assuming in the extracellular matrix, you wouldn't need to
So I can give you
that.
additional insights. Okay.
Okay.
Myofibroblast are the cells which is associated with the resolution of a wound. If myofibroblast are higher, that means the wound is more or less completing the healing process. Okay.
Okay.
And the myofibroblast marker is alpha SMA, smooth muscle alpha actin, which is a cytoplasmic protein.
Oh,
Mm.
okay.
Yeah,
So we're
yeah, yeah.
dealing with the cytoplasmic protein. Okay. So
Yes.
then I guess you would then treat the cells, put in the suspension buffer, and then um permeabilize the cells so that you can access those cytoplasmic proteins,
Hmm.
and then run it through flow cytometry. So I don't know if you would put like an antibody for those cells, I mean for the protein, so that or like a fluorescence, so that the flow cytometry can identify the individual cells. And I'm assuming based on the size of the cells, it would then end up you would then see. I mean, you'd be counting. You're not looking for complexity, right? So you're just counting forward scatter in this case.
Yes.
I'm presuming, yeah.
Yeah, yeah,
Um,
yeah. You are right.
like,
I can repeat the process again as you mentioned. You are right. So first, we need to isolate cells from the wound. Then we need to make a cell suspension. Since the protein is cytoplasmic, we need to definitely blocking step is there before that. We will be blocking all the unwanted antigens. Then we will be fixing and permeabilizing the cell. We will be using a fluorochrome tagged antibody that will go and inside the bind the cytoplasmic protein. We will do the flow cytometry, and based on the fluorescence, we will quantify the cells and do the analysis. Yes, that's the workflow. You are right. Okay, Kristen, you are next.
Okay. Um, I would do the fourth one.
Oh
Um,
yeah, fourth one.
yeah, so GFP, you would use transfection, which is like taking a plasmid that you designed, which would have like GFP, like the DNA sequence for GFP in it. And
Mhm.
then you would transfect that into the cells that you're studying, with like electroporation or some method for transfection. And then now that that like GFP marker is in your cells, like nucleus, I guess, or like in their DNA now, um, that itself is a fluorescent protein. So you could add another marker though, like another antibody stain if you wanted to like isolate certain kinds of cells, I guess. I don't know what cells we transfected in this. Do you want to give me an example?
Okay, okay. So for example, I transfected my stem cell, okay, with protein X. Okay. So I am anticipating, okay, the transfection is successful and the cells are expressing protein X. So protein X is tagged with GFP. That means if we have a fluorescence, that will simply demonstrate the expression of protein X.
Okay.
Can you elaborate this now?
Yeah, so then maybe I'd want to add, so now I have these transfected
Okay,
cells.
forget about all those transfection procedures and everything. So you are running a flow cytometry core, and I did the experiment, and I
Yeah.
am giving you my samples, and I am telling, okay, I want to quantify the expression of GFP positive cells.
Yeah, so I would take the cells and add any other antibodies if you wanted to, and
Mm-hmm,
then like use a fix and permealize that.
mm-hmm,
Um, and then like wash it with PBS and then take it to go flow and
mm-hmm,
like the GFP itself would appear on the
mm-hmm.
machine and you could like gate for that, I guess.
Yes, you are right. So, this process would be very simple. Just take the simple suspension and run the flow.
Okay, no fix and perm.
Nothing, because our intention is to see GFP positive cells. Okay, if, for example, if I have 100,000 cells, my transfection efficiency is 90 percentage. So
Mhm.
just load that 100,000 cells, and you will get 90,000 cells positive for GFP and 10,000, non-GFP or non-fluorescent cells. Okay, that's a very simple experiment. If you have any experiments like this, simply take the cell suspension and run it as such. Okay, and again, depends. So suppose if I want to check the co-expression of another molecule, CD90, for example. If I want to check the co-expression of CD90 with my GFP positive cells, definitely I can use another antibody. It all depends on how we plan our experiments, what outcome we are looking forward. Okay, I think anyone want to go other assignments, or if you are clear?
Question. So
Yes.
why wouldn't you want to permeabilize with GFP again?
GFP, usually what we do GFP, if you remember the experiments which we did in our lab with CRISPR, you know that? Have you seen that?
Yes.
So,
Yeah.
yeah, in that CRISPR technology, we will be either overexpressing or silencing a gene, for example. Okay. So when we, in the case of gene overexpression, we will be using GFP protein along with our protein of interest. Here, my protein of interest is protein X. That protein X DNA sequence will be tagged with GFP. GFP is green fluorescent protein. Okay, so whenever the transfection occurs, and if the transfection is successful, whenever protein X is getting expressed, along with that, GFP will be there. Okay, so if the transfection is successful, if we look the cell under microscope and uh, give a blue laser or blue light, it will give a green fluorescence. Okay, okay, so that means these two proteins are expressing together, and we will be growing those cells for generations, and we need to see if protein X is there. Just simply run a flow cytometry with GFP alone, because GFP is inherently present in that transfected cells. Hope you got my point.
Yes. So because it's not in the cytoplasm at all or in the nucleus, it's just a protein that's being expressed. I see.
Yes, yes, yes.
So,
Yeah, yeah. We can make it express anywhere. So suppose the protein X is a nuclear protein, it will express in nucleus. If protein X is a cytoplasmic protein, it will express in the cytoplasm. Or if protein X is a membrane protein, it will express in the membrane. So wherever our protein X is expressing, along with that, GFP will be coming out too. Okay. Okay, any further questions?